First, I'd like to thank the organizers for this fantastic meeting. I think it's one of the best cancer meetings I've been to yet. And it's just been really interesting with respect to metastasis. So what I'll tell you about today is a process we discovered that drives most epithelial cell death called extrusion here. Let me just get my pointer. Extrusion, and how that can be hijacked by cancer cells to instead cause invasion, which is what you see in this movie here, which we'll get back to later. And how we came across this was really by accident. We were looking, we're epithelial cell biologists in the main, and so we were really interested in just how do all these epithelial cells work tightly together to make a collective barrier for all of our organs. And what we were, this looks pretty straightforward if you're just looking at this picture of an epithelium where it just looks like bricks in a wall. But then if you consider what we know is that most epithelia turn over at some of the fastest rates in the body, with I think the gut really taking the prize at hundreds of billions per hour. And so if you think about that, the entire gut lining is turning over within 3 to 4 days. So this is a lot of cell division and cell death. And it really brought to mind some of the following questions. One is, how does this barrier cope? How does it, and also your epithelium has these different functions that are critical to organs, each specific organ, but the main thing is that they all have in common is keeping this barrier. So how is it able to maintain this barrier in the face of all this cell death and division? And then the other thing we were very interested in is what, I'm not sure if you can see what some of my lines, my face is in the way. What connects cell division to cell death? And you can imagine that every time on average a cell divides, another cell will have to die. And if you didn't do this correctly, you could just lose all of your cells and your barrier and organ function. Or on the other hand, if you get too many cells dividing and not enough dying, you could cause tumor formation. And this is, as we all know, epithelia, simple epithelia like these are the sites where most solid tumors form. So this is very relevant to epithelia. And so we thought that this was an interesting question to address. And really surprisingly, when we got to this field, it was just like people studying cell death in separate labs to those that are studying cell division, going to separate meetings, not really talking to each other, and thinking about cell death and division really in response to some kind of stimulus. So, some kind of growth factor or apoptotic stimulus. And we didn't have a very good idea of just how this happens normally. And we thought that's important to understand, because once we, if we understand how it goes on normally, then we'll start to understand how it doesn't happen in abnormal situations as disease. And what this boiled down to, which is actually so simple that a 10-year-old could have figured this out, is that it's mechanics. So it's just simple mechanics. If there's not enough cells, the stretch that all these cells experience as they're holding together rapidly causes cells to divide. And on the other hand, if there's too many cells, the crowding forces cause these cells to die through this process of cell extrusion that we discovered when I was a postdoc here at the LMCB. And so in this process, a cell that's fated to die, but still alive, actually, importantly, still alive, will contract a cable of actin and myosin around it, the neighboring cells and the cell that's extruding, and extrude out into the lumen, where it then goes on to die. And by doing this, they're able to keep constant cell numbers, approximate constant cell numbers. And there's actually regions that are more likely to divide and regions that are more likely to extrude, as we know in the gut. So in the crypt, you might see cells dividing. In the villus, you see cells extruding. And as since we've had this discovery of extrusion, others have gone on to identify that this is a conserved process that happens from sea sponge to human. So it just seems that all epithelia that we've investigated have this conserved process of extrusion that really is driving, in a fundamental way, cell death. And it is probably for this reason that it's having new roles in that other people are investigating in bacterial and viral pathogenesis, development, cell competition. We're also looking at asthma, and what I'll talk about today, obviously, is cancer. And so what we found is that, you know, if we understand that extrusion is causing a lot of this epithelial cell death, then it stands to reason if there's too much of it, you may have problems with your barrier function. And that's what we're finding in asthma, is that too much crowding coming from a bronchoconstrictive event can cause too much extrusion that causes this disruption in the barrier function and inflammation. And on the other hand, what we found was by identifying the signaling that drives this process, that we realized that it gets misregulated in some very aggressive tumor types. And so instead of sending these cells out to die, they'll form masses, but then they'll also cause this weird aberrant type of extrusion that we call basal cell extrusion, which I'll show you today can cause cells to invade. So what I said is a lot of this comes from our discovery, what signals extrusion. And what I'll tell you just simply, this is a whole other talk I could give. It's complicated, obviously, and there's a lot at play, but just to give you the main take-home point is that what's really critical is production of a lipid signaling ligand, sphingosine phosphate, activated by sphingosine kinase. This can be activated, importantly, in response to apoptotic stimuli or the crowding, which I mentioned before, and the player here is got the Nobel Prize, piezo-1. So the crowding is sensed by piezo-1 that activates sphingosine kinase activation through calcium signaling to produce this lipid ligand. And then it binds a G protein coupled receptor called sphingosine phosphate 2. This then activates Rho and RAC to cause contraction around and below that squeezes the cell out of the layer. Now, importantly, what we found is that just by investigating what would happen if we disrupted this pathway. So we were using zebrafish, which we'll be using throughout most of the talk here, as a really nice epithelium that we can see live everything that's going on. So this is the fish, zebrafish embryonic tail, and you can see this is made of a bilayer that's just this nice epithelium that coats the whole embryo, obviously, but here it's just pure epithelium. And if we looked in a mutant, we thought, well, a mutant in sphingosine phosphate 2, the lipid ligand receptor, would probably cause holes because these cells wouldn't extrude anymore. What we were surprised to find is not only does it destroy the barrier function, but you get very rapidly masses forming at sites where these cells should have extruded. This is within only 4 days of not having this receptor. So this was interesting. And Yapeng Gu, the postdoc in my lab who identified this pathway, who is a doctor, went on to think, well, maybe there's something wrong in cancer. Why don't we look? And so just going through OncoMine, we found that all pancreatic cancers and some types of lung and colon cancer also lack this sphingosine phosphate 2 receptor. So it's vastly downregulated in these cancers. And so to test whether that was actually had any role or was just a bystander effect and just coincidence, he used an orthotopic pancreatic cancer mouse model. So he took pancreatic cancer cells, labeled them with GFP, and then you inject them into the pancreas, and they'll grow into these tumors that fill up the entire cavity of this mouse here. I'm just showing you the whole cavity. You can see it's filled with these metastatic cancer cells here. But if you only replace the sphingosine phosphate, so if you just put in sphingosine phosphate labeled to GFP, then you drastically reduce the size of the tumors and the metastases. We only found 1 metastase in 10. So this was really suggesting that it wasn't just playing a bystander role, that maybe controlling extrusion, which these cells that now they have sphingosine phosphate 2 rescued could also extrude properly, would reduce this tumor growth. Since this finding that sphingosine phosphate 2 is important for extrusion signaling and preventing this tumor formation and metastasis, we found that not only pancreatic cancer regulates the sphingosine phosphate, but that other oncogenic mutations that drive quite aggressive cancers, such as APC and KRAS B12 mutations, they convergently attack this pathway. So in this case, they misregulate the trafficking of this lipid ligand instead of the receptor. And again, in all these cases where this extrusion signaling is disrupted, and it's interesting because they do it in sort of different ways, but they all kind of converge on the same pathway. What we find is the same things, is that you get masses forming at sites where these cells should have extruded and died. And then you also get this part that I was mentioning, which is that these cells can also extrude in an aberrant way basally back into the tissue that the epithelia encases. And this we're now showing can initiate invasion and metastasis. And so the way that we did this, we could first see this in 3D cysts and organoids. This was quite clear, but we really wanted to see this live and see what would happen to these cells in an in vivo system. And so to do that, John Fadul joined my lab and started developing the zebrafish as a model for simple epithelia, the zebrafish epidermis, sorry, as a model for simple epithelia where tumors form. And by doing this, we could actually see all these events live. So what he did simply was, at the very early one cell stage, inject these embryos with either a KRAS tethered to GFP, which we know disrupts extrusion, as I mentioned before, or GFP tethered to a membrane, CAX here. And what you can see pretty readily is that in the case of the CAX, it's just these cells kind of overtake the whole epidermis. But in the KRAS, there's fewer of them, which is sort of interesting. But the ones that are there are forming these masses, as we saw previously in the other zebrafish, where we blocked the pathway of the sphingosine-1 phosphate 2 mutant. Now, what gets more interesting is if you hone in on these and image them live, which is the whole point of using the zebrafish in the first place, you can see that the control cells just sort of sit there. They'll divide and take over the epidermis. But in the KRAS cells, in fact, they're more likely to extrude, and you can see this one here extruding. And when they do, they preferentially extrude basally back into the tissue that this epidermis is encasing. So one thing that was sort of surprising to us here was that the places where this happened were at separate sites to where, if I can close this so I can actually point to things. We're at separate sites where the masses formed. So the masses were forming at sites where the cells typically extruded more at the curved edges and older, where the cells were sort of older and convergently migrating to. And the places where the cells were invading by basal extrusion were places that were typically never extruding. They were usually places where they were dividing instead. And these cells seem to have a lot more proliferative advantage as well. So this has suggested that these two activities were quite separated, that if you were older, maybe you'd form masses, but if you were younger, you would be preferentially extruding basally. And this just shows you a good example of this whole thing happening. So here you can see just a small mass forming here that's circled. But if you look right here, you can see a cell that's about to invade by this extrusion. And so what you'll notice is a sort of explosion event, and then the cell gets released underneath the epidermis. So there's an explosion, and now the cell, I've tracked it, is migrating throughout the whole embryo through the duration that we filmed, and potentially longer because these cells seem to stick around. So we wondered, as we were watching this, what this explosion was. It was kind of confusing to us because we kept seeing this explosion. And when we investigate this by just looking at a little higher resolution imaging of this, what we noticed is that every time these cells were invading, they simultaneously pinched off their entire apical domains here. So you'll see this in this situation. This is pinching off this entire apical domain, and then the cell emerges out underneath. And what you'll see is that it's stripping off the KRAS, which is GFP tethered. And so sometimes we were losing these cells because they were at this apical domain. What's important to note here is that as these cells were invading, they seem to be stripping off their E-CADHERIN and other apical surface epithelial determinants, which there are many at the apical surface, because when we stained for KRAS when we stained for E-CADHERIN of these cells that had internalized, there was just none of the cells out of many of these cells. So I'm just going to see if I can undo my laser pointer so I can, there we go. I can see what I'm doing. Sorry about that. So all of these lacked E-CADHERIN at ALF 340 that we measured. So after these cells were released, they had a lot of predictable behavior. So we could see them, these invading cells, migrating throughout the whole, throughout the whole embryo, as you saw before. We frequently see them dividing. We also could see them very readily getting into the bloodstream. And then we also saw them adopt a variety of different cell fates that were sort of surprising to us. So what we felt like is that these cells, as they're invading, are pinching off and becoming partially dedifferentiated. And we could almost call it an epithelial to mesenchymal transition, but not all the cells, in fact, were becoming mesenchymal. We found if we stained for a mesenchymal marker SNAIL1B and by RNA in situs, or with an antibody to ENCADHERIN, we could see that some of the cells were adopting mesenchymal phenotypes, but in fact, it was only a portion of these cells that were doing so. And now work from Teresa Zueta-Corabarasa in my lab, who choose following up this phenotype of mesenchymal cells seems to be very tightly linked to how much contortion these cells experience. And if you look at this movie, you can see that as the cell is migrating, it's getting very contorted, so much so that if we looked at the nuclei, we could see that it was causing the nuclei to get just smashed as it was going through. And so we're wondering now if this can be something that maybe is promoting, DNA, lots of DNA, mechanical DNA damage, and maybe helps promote the dramatic changes, genetic changes that you may be selected for during metastasis. So this is something that we hope to be publishing quite soon. The other cell type that we saw were these cell types that we call neuronal-like. We don't know, they would just kind of migrate throughout the whole body of the embryo. And sometimes they would migrate along previously established neurons. Sometimes they look like they co-localize with neurons. So we're not quite sure what these cells are, but they were quite interesting and very frequent. So all of these cell types that we saw, all of these behaviors were quite interesting, but in the backdrop of a wild type background, so it's just KRAS alone, by 5 days post-fertilization, almost all of them had died. There might be a few hanging out here and there, but really they're mostly gone. And that sort of makes sense to us because we know one mutation does not cause a tumor. But if we looked with a common collaborating mutation in P53, we found that now these cells could survive and form metastases throughout the body. Mostly we saw them in the head, but we could see them really just explode into these tumors. So it really suggests to us then that these cells, they can invade very readily, but many of them don't really survive. And maybe it's through all this contortion and damage that they're experiencing. But when you have a P53 mutation, it now allows those cells to survive and form these metastases. So what we think then is that this basal extrusion can cause invasion and metastasis, but this is all from a zebrafish epidermis. And we're very aware of this, as are many people who've criticized the zebrafish, that this is all good and well in the zebrafish, and could this be true in our own bodies, in the mammalian tissues? And so just to get the first glimpse of this, what we thought we'd do is turn to another model system we're using in my lab quite a bit for the asthma, is the mouse lung epithelia. So we've been using these mouse lung slices to mimic the bronchoconstriction. You can see this here. You can see all these cells extruding. And we were really impressed with how easy they are to image. We can keep them in culture for up to, I think, 3 to 4 weeks. We haven't gone past that, but it seems like they're surviving quite well. And so what we decided to do, what John is doing now with Will Hill, who's in the Swanton lab, is to use another KRAS model in the mouse that, and so far we just KRAS, not the P53. And then what's beautiful is that we can use adenovirus to just make these express mosaically and in any tissue type, any type of epithelium that we like. Then we sacrifice the mice, harvest the lungs, make the lung slices, and then use spinning disc microscopy to film this. And so what we're quite gratified to see is that we're seeing very similar findings. I apologize for some of the earliness of these. These are kind of hot off the press, but we're seeing this now. These cells are labeled, the ones that are KRAS are in pink here, or in white is overlapping here. And you can see them start to migrate and sort of pinch off from their original spots, we think in the same mechanism. And what's kind of amazing too is we can see these cells start to invade, and they seem to be migrating along the alveoli. Sorry, this is fading a bit as we're going along, but if you see that again, you can sort of see it's taking the tracks of these alveoli. So it'll be quite interesting to see how these work, but it looks so far that we are replicating the same findings in the mice, and we're hoping also to go back to some human organoids. So we think that this finding really it has some similarities, but it is also at odds with the main classical models of metastasis. This one is for pancreatic cancer, which we think is relevant here, which suggests that the KRAS mutations really allow the cells to form primary tumors, which, yes, we saw that too. But what it assumes is metastases come necessarily from these primary tumors. And what we think is maybe that's not really the case. In our hands, it's almost as if the primary tumor is over here saying, look at me, making a distraction. Meanwhile, cells can be invading at distant sites by this basal cell extrusion. And this basal cell extrusion, importantly, is stripping off simultaneously, mechanically, the apical epithelial determinants. Now the cell can be released to create a sort of de-differentiated state. These cells can then go on to form these different cell types, which we're interested to see whether they're relevant to humans and even mice, and also get into the bloodstream. But again, they'll only form metastases if they lack a mutation in something like P53, which would allow it to survive. So we think that just this new model is leaving open questions to for the future. One is this early independent loss of these cells invading, but also surface epithelial markers. Maybe we're losing some of the critical components of the tumor by not looking at different markers that might be in cytoplasmic epithelial markers. Secondarily, it suggests that we could maybe screen very early on for cells that are not really cancerous, but have metastatic, they're like metastatic precancerous sort of cells. So maybe we need to think about the way we're doing this. Then potentially we could just use preventative medicine. So I can imagine screening for epithelial cells, maybe transformed, not really cancerous, and using something that's not so toxic to prevent metastases rather than treat them after they've formed. And finally, this finding that we have that contortion is causing this migration. What we're thinking is that potentially this could cause either the cells to die, which would be good, but the ones that don't die and maybe have this P53 loss may take on new fates and become stronger in sort of a Nietzsche-esque way. So these are just sort of open ideas for question based on what we found in the zebrafish, but we still need to find in the mammalian systems. So I'd like to end by just thanking my lab, my growing lab. It's been also nice. We've moved here like about a year before lockdown, so it's been really nice to get to know some of you, and that sort of put thwarted our efforts to get to know people in the cancer community here. But I'd really like to especially thank John Fadul and Teresa, who've been working on this project previously, and funding sources. Thank you.